Class 11, Chapter 8, Cell, the Unit of Life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of basic unit of life, the cell. In all the living organisms, all organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. 8.1 What is a cell? Unicellular organisms are capable of independent existence and performing the essential function of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structure and functional unit of living organism. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structure detail of the cell. 8.2 Cell theory. In 1838, Matthias Sheldon, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which form the tissue of the plant at about the same time. Theodore Schwann, 1839, a British zoologist, studies different types of animal cells and reported that cells had a thin outer layer, which is today known as the plasma membrane. He also concluded, based on his studies on plant tissue, that the presence of cell wall is a unique character of the plant cell. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cell and products of cell. Sheldon and Schwann together formulated the cell theory. The theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divide and new cells are formed from pre-existing cells. Omnicellular, e-cellular. He modified the hypothesis of Sheldon and Schwann to give the cell theory a final shape. Cell theory, as understood today, is: first, all living organisms are composed of cell and products of cell. Second, all cells arise from pre-existing cell. 8.3 An overview of cell. You have earlier observed cell is an onion peel and or human-shaped cell under the microscope. Let us recollect their structure. And the onion cell, which is a typical plant cell, has a distinct cell wall and as its outer boundary, and just within it is the cell membrane. The cell of a human cheek have an outer membrane as a delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material DNA. Cells that have a membrane bound nuclei are called eukaryotes, whereas cells that have a lack membrane bound organelle are called prokaryote. In both prokaryote and eukaryote cell, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plants and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Beside the nucleus, the eukaryotic cell have other membrane-bound distinct structures called organelles, like the endoplasmic reticulum, the Golgi complex, lysosome, mitochondria, microbodies, and vacuoles. The prokaryotic cell lacks such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plant and mitochondria and on rough Animal cell contain another non-membrane bound organelle called centrosome, which help in cell division. Cells differ greatly in size, shape, and activities. Figure 8.1. For example, mycoplasma, the smallest cell, are only 0.3 μm in length, while bacteria could be 3 to 5 μm. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7.0 μm in diameter. Nerve cells are some of the longest cells. Cells are also greatly in their shape. This may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Figure 8.1 diagram showing different shape of the cell. 8.4 Prokaryotic cell. The prokaryotic cell are represented by bacteria, blue-green algae, mycoplasma, and PP yellow. Pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cell. Figure 8.2. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus spherical, vibrio comma-shaped, and spirillum spiral. The organism of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shape and function. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmid. The plasmid DNA confers certain unique phenotypic character to such bacteria. One such character is resistant to antibiotic. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryote. No organisms like the one of the eukaryote are found in prokaryotic cell except for ribosome. Prokaryotes have something unique in the form of inclusion. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryote. They are essential in folding of cell membrane. 8.4.1 Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cell, have a chemical complex cell envelope. This cell envelope consists of a tightly bound three-layer structure. The outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of difference in the cell envelope and the manner in which they respond to the staining procedure developed by Gram-Wise. Those that take up the Gram stain are Gram-positive, and the other that do not are called Gram-negative bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be a loose sheath called slime layer in some, while in other it may be thick and tough called capsule. The cell wall determines the shape of the cell and provide a strong structure support to prevent the bacteria from bursting or collapsing. The plasma membrane is selectively permeable in nature and interact with the outside world. The membrane is similar structurally to that of eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. They help in cell wall formation, DNA replication, and distribution to daughter cell. They also help in respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extension into the cytoplasm called chromatophore, which contain pigment. Bacterial cell may be motile or non-motile. If motile, they have thin filament extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Beside flagella, pili and fimbriae are also surface structure of the bacteria but do not play a major role in motility. The pili are elongated tubular structure made of a special protein. The fimbriae are small bristle like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rock in streams and also to host tissue. 8.4.2 Ribosome in incision bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together from 70S prokaryote. Ribosomes are the site of the protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. The ribosome of a polysome translates the mRNA into protein. Incision bodies, reserve material in prokaryotic cell, are stored in the cytoplasm in the form of incision bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granule, synophysae granule, and glycogen granule gas vacuole are found in blue, green, and purple and green photosynthetic bacteria. 8.5 Eukaryotic cell. The eukaryote include all the protist, plant, animal, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nuclei with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. The genetic material is organized into chromosomes and eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell wall, plastid, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. Figure 8.3. Figure 8.3 diagram showing a plant cell, B animal cell. Let us now look at individual cell organelle to understand their structure and function. 8.5.1 Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cell RBCs, enable the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and protein. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail towards the inner part. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. Figure 8.4. In addition to phospholipid membrane also contain cholesterol. Later biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipids vary considerably in different cell type. In human being, the membrane of the erythrocytes has approximately 52% of the protein and 40% of the lipid. Depending on the ease of extraction, membrane protein can be classified as an integral and peripheral. Peripheral protein lie on the surface of membrane, while the integral protein are partially or totally buried in the membrane. Figure 8.4 Fluid mosaic model of plasma membrane. An improved model of the structure of cell membrane was proposed by Singer and Nichols in 1972, widely accepted as fluid mosaic model. Figure 8.4. According to this, the quasi-fluid nature of lipid enables lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation of intercellular junction, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecule across it. The membrane is selectively permeable to some molecule present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecule cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example: Na plus K plus pump. 8.5.2 Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only give shape to the cell and protect cell from mechanical damage and infection, it also help in cell-to-cell interaction and provide barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, calectins, mannese, and mineral like calcium carbonate, while in other plant it consists of cellulose, hemicellulose, pectin, and protein. The cell wall of a young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell mature, and the secondary wall is formed on the inner toward membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which hold or glues the different neighboring cells together. The cell wall and middle lamella may be transferred by plasmodesmata which connect the cytoplasm of neighboring cells. 8.5.3 Endomembrane system. While each of the membranous organelle is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their function are coordinate. Their endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, and vacuole. Since the function of the mitochondria, chloroplast, and peroxisome are not coordinate with the above component, these are not considered as part of the endomembrane system. 8.5.3.1 Endoplasmic reticulum Electron microscopic studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum Figure 8.5. Hence, divides the intercellular spaces into two distinct compartments: luminal inside and extraluminal cytoplasm compartment. The often show ribosome attachment to their outer surface. The endoplasmic reticulum bear ribosome on their surface called rough endoplasmic reticulum (RER). In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum (SER). RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of protein. In animal cell, lipid-like steroidal hormone are synthesized in SER. 8.5.3.2 Golgi apparatus. Camilo Golgi 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 mm to 1.0 mm diameter. Figure 8.6. These are stacked parallel to each other. Varied number of cristae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and the trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging material to be delivered either to the intracellular target or secreted outside the cell. Material to be packaged in the form of vesicles from the fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remain in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of the formation of the glycoprotein and glycolipid. 8.5.3.3 Lysozyme. These are membrane-bound vesicle structure formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all type of hydrolytic enzyme: hydrolase, lipase, protease, carbohydrase. Optimally active at the acidic pH, these enzymes are capable of digesting carbohydrate, protein, lipid, and nucleic acid. 8.5.3.4 Vacuole. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other material not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cell, the vacuole can occupy up to 90% of the volume of the cell. In plant, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protist, food vacuole are formed by engulfing the food particle. 8.5.4 Mitochondria. Saying mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. In terms of shape and size, also considerable degree of variability is observed. Typically, it is a sausage-shaped or cylindrical, having a diameter of 0.2 to 1.0 mm, average 0.5 mm, and length 1.0 to 4.1 mm. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, the outer compartment and the inner compartment. The inner compartment is filled with the dense homogeneous substance called the matrix. The outer membrane forms the continuous illuminating boundary of the organelle. The inner membrane forms a number of enfolding called the cristae. Crista towards the matrix. Figure 8.7. The cristae increase the surface area. The two membranes have their own specific enzyme associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of the ATP, hence they are called powerhouse of the cell. The matrix also possess single circular DNA molecule, a few RNA molecule, ribosome 70S, and the components required for the synthesis of protein. The mitochondria divide by fission. 8.5.5 Plastids. Plastids are found in all plant cells and in euglenoid. These are easily observed under the microscope as they are large. They bear some specific pigment, thus imparting specific colors to the plant. Based on the type of pigment, plastid can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigment which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat soluble carotenoid pigment like carotene, xanthophyll, and other are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastid of varied shape and size with stored nutrients. Amyloplasts store carbohydrates, starch, example potato. Elioplasts store oils and fats, where the aleuroplasts store protein. Majority of the chloroplasts of the grain plant are found in the mesophyll cell of the leaf. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelle having variable length 5 to 10 mm and width 2 to 4 mm. The number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40 per cell is the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoid are present in the stroma. Figure 8.8. Thylakoid are arranged in the stacks like the piles of the coins called grana. Singular granum are the integranal thylakoid. In addition, there are flat membrane tubules called the stroma lamellae connecting the thylakoid of the different grana. The membrane of the thylakoid encloses a space called a lumen. The stroma of the chloroplast contain enzyme required for the synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecule and ribosome. Chlorophyll pigment are present in the thylakoid. The ribosome of the chloroplast are also smaller 70S than the cytoplasmic ribosome 80S. 8.5.6 Ribosome are the granular structure first observed under the electron microscope as dense particle by Gregor Palade 1953. They are composed of ribonucleic acid, RNA, and protein and are not surrounded by any membrane. The eukaryotic ribosome are 80S while the prokaryotic ribosome are 70S. Each ribosome has two subunits, larger and smaller subunit. Figure 8.9. The two subunits are of 80S ribosome and 60S and 40S, while that of 70S ribosome are 50S and 30S. Their S Swedberg's unit stand for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosome are composed of two subunits. 8.7.5 Cytoskeleton, an elaborate network of filamentous proteinaceous structure consisting of microtubule, microfilament, and intermediate filament present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. 8.5.8 Cilia and flagella. Cilia, single coelom, and flagella, single flagellum, are hair-like outgrowth of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of the cilium or the flagellum show that they are covered with plasma membrane. Their core, called the axon, possesses a number of microtubules running parallel to the long axis. The axonum usually has 9 doublets of radially arranged peripheral microtubule and a pair of centrally located microtubules. Such an arrangement of axonal microtubule is referred to as 9 plus 2 array, figure 8.10. The central tubule are connected by the bridges and is also enclosed by the central sheath, which is connected to one of the tubule of each peripheral doublet by a radial spoke. Thus there are 9 radial spokes. So their peripheral doublet are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. 8.5.9 Centrosome and centriole. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar material. Both the centriole and a centrosome lie perpendicular to each other, in which each has an organism like the cartwheel. They are made up 9 evenly spaced peripheral fibrils of tubulin protein. Each of these peripheral fibrils is a triplet. The adjacent triplet are also linked. Their central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubule of the peripheral triplet by radial spokes made of protein. The centriole form the basal body of cilia or flagella, the spindle fiber that give rise to spindle appendages giving during cell division in animal cells. 8.5.10 Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dye was given by the name chromatin by Fleming. The interface nucleus of the cell when it is not dividing, has the highly extended and elaborate nucleoprotein fiber called chromatin. Nuclear matrix fibers and one or more spherical bodies called nuclei, sing nucleolus, figure 8.11. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remain continuous with the endoplasmic reticulum and also bear ribosome on it. At a number of places of the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are two passages through which movement of RNA and protein molecule take place in both directions between the nucleus and cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. Example: erythrocyte of many mammals and sieve tubes of cell of vascular plant. Would you consider these cells as living? The nuclear matrix or the nucleoplasma contain nucleoplasm and chromatin. The nuclei are spherical structure present in the nucleoplasm. The content of my nucleolus is continuous with the rest of the nucleoplasm as it is not membrane bound structure. It is the site for active ribosomal RNA synthesis. Large and more numerous nuclei are present in cell actively carrying out protein synthesis. You may recall that the interface nucleus has a loose and distinct network of the nucleoprotein fiber called chromatin, but during different stages of cell division, cells show structured chromosome in place of the nucleus. Chromatin contain DNA and some basic protein called histone and some non-histone protein and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 23 pairs of chromosomes. You will study the detail of DNA packaging in the form of chromosome in class 12th Every chromosome visible only in dividing cell essentially has a primary construction or the centromere, on the sides of which disc-shaped structure called kinetochores are present. Figure 8.12. Centromere hold two chromatids of the chromosome based on the position of the centromere. The chromosome can be classified into four types. Figure 8.13. The metacentric chromosome has middle centromere forming two equal arms of the centrosome. The subset of metacentric chromosome has a centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming the extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary construction at a constant location. They give the appearance of a small fragment called satellite. 8.5.11 Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal. Summary: all organisms are made up of cell or aggregate of cells. Cells vary in their shape, size, and activities, functioning based on the presence or absence of membrane-bound or nucleus and other organelles. Cells and hence organism can be named as eukaryotic and prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Plant cell have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitate transport of cellular molecule. The endomembrane system include Golgi complex, lysosome, and vacuole. All the cell organelles perform different but specific functions. Centrosome and centriole form the basal body of cilia flagella that facilitate locomotion. In animal cell, centriole also forms spindle apparatus during cell division. Nucleus contain nuclei and chromatic network. It is not only control the activities of organelles but also plays a major role in heredity. Endoplasmic reticulum contain tubule or cisternae. There are of two types, rough or smooth. Helps in the transport of substance, synthesis of composed protein, lipoproteins, and glycogen. The Golgi body is membranous bound, composed of latent secretion of cells are packed in them and transported from the cells. Lysosome are single membrane structure containing enzyme for digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasmic or are associated with Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate. They are bound by double membrane. The pigment containing organelle found in plant cell only. In plant cells, chloroplasts are responsible for trapping light energy, responsible for photosynthesis. The grana in the plastid is the site of light reaction and the stroma of dark reaction. The green colored plastid are chloroplasts which contain chlorophyll, whereas the other colored plastid are chromoplasts which may contain pigment like carotene and xanthophyll. The nucleus is enclosed by a nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. The cell is the structure and functional unit of life.